Retina Rounds podcast number 19. As part of our Giants in Retina series, Retina Rounds is honored to welcome a true giant in biomedical innovation, Dr. Mark Humayun. Dr. Humayun is Professor of Ophthalmology and co-director of the University of Southern California Roski Eye Institute and director of the USC Dr. Allen and Charlotte Ginsberg Institute for Biomedical Therapeutics. Dr. Humayun is an ophthalmologist, engineer, scientist, and inventor, and has the distinction of being the only ophthalmologist ever to be elected a member of both the U.S. National Academies of Medicine and Engineering. Dr. Humayun co-invented the Argus series retina implants and has more than 125 patents. In 2015, for all his contributions to biomedical innovation, President Barack Obama named Dr. Humayun a recipient of the National Medal of Technology and Innovation. In today's podcast, Dr. Humayun talks about his journey to ophthalmology and biomedical innovation and provides pearls on how to bring an innovative idea to reality. Let's check it out.
So, welcome everybody to this week's podcast of Retina Rounds. This week we have a giant in retina joining us, Professor Mark Humayun of the University of Southern California. Mark is a distinguished professor of both ophthalmology and engineering at the University of Southern California and has made some of, if not the most impactful innovations that have translated into instruments that we as vitreoretinal surgeons use every day. And so I'm looking forward, Mark, to talking about your journey into innovation and how you navigate the complex interface between invention and reality. So thanks for joining us, Mark.
Oh, thanks for having me, Steve.
It's a great pleasure. And Mark is, I'm proud to say, one of my old dear friends, and he routinely humiliates me on the tennis court. And a little birdie told me that he recently won a rather big tournament at a local facility. So tell us about that, Mark. That was, I'm sure it was a 5 tournament or an open.
Well, yeah. Well, not much to brag about. My good tennis days are behind me, as they are for most of us at our age. But it was a fun. It was just a local club match, so it was fun. Nothing too much to talk about. But it's a good day, Steve, when you and I can go out there and hit the ball and not get hurt, right?
That's exactly right. That's exactly right. So, Mark, tell me about how you landed in ophthalmology. What was your journey into medicine and then ophthalmology?
Well, it wasn't clearly straightforward. Ophthalmology is something that we don't get a lot of exposure to in medical school, and that was the case when I was at Duke. There wasn't a lot of exposure, and initially I was interested in going into neurosurgery, and fortuitously, while I was doing the 1 year away from Duke at Johns Hopkins, I ran into um, Neil Miller, who's a world-renowned neuro-ophthalmologist, who was in the operating room with Don Long, who was the chair of neurosurgery, who was my mentor, who I was spending the year with, and I really liked those aspects of ophthalmology and neuro-ophthalmology, and wanted to explore ophthalmology more. And at that time, at the same time, my grandmother started to lose vision because of diabetic retinopathy. So, my grandmother raised me, and so it was very personal. So, both from a professional aspect of being intrigued by neuro-ophthalmology and ophthalmology, as well as on a personal level because of my grandmother, it allowed me to think about, focusing in ophthalmology. And that's where I sort of changed my course and went into ophthalmology.
And were you an engineer by training before you went to medical school?
That's interesting. I had a lot of math, but I would say no, I was not really an engineer by training, and it was through the project. A lot of the things that we developed, and in particular the retinal implant project, bioelectronic implant, the Argus 2, it required so much engineering that I had to go back and do a PhD in engineering at the same time that I was a resident at Duke University Eye Center. So it was something that came later, and it came out of necessity of the areas of interest that I had, from instrumentation to that program, that really made me go back and get my PhD. But luckily, I did have a lot of the maths background, because without that, it wouldn't have been possible.
Right. And I'm not even sure I knew this, you conceived and started working on the Argus 2 while you were a resident. And as a result of the needs of the project, rather than find engineers to collaborate with, you rolled up your sleeves and went and got a PhD. And do I remember you got the PhD from Georgia Tech or was it from Duke?
Well, you're right. It was out of necessity of getting the PhD. I did try to collaborate with engineers, but it was like we're talking past each other. They didn't understand anything about sodium, potassium, and cells, and I didn't understand anything about a PN junction or a transistor. So it was just a very difficult type of situation, and so communications were not getting through. The project was being slowed, but because of the project, I ended up getting the PhD. And actually, the PhD was interesting. I was able to take courses at Duke, North Carolina State, and University of North Carolina at Chapel Hill, and because each one had different strengths. Duke in biomaterials, North Carolina State in solid state, integrated chip circuits, and University of North Carolina Chapel Hill. I had a lot of bioinformatics, and again, a lot of packaging for electronics. But long story short, my degree officially is from the UNC Chapel Hill, but I got to take courses from all those three universities.
Oh, that's a fantastic story. And then when you finished your training at Duke in ophthalmology, where did you go next?
So there, I had a choice to stay on at Duke, and that was clearly, one of the premier, and still is one of the premier vitreoretinal fellowships. With Then Dr. Mockermer was still very active. Brooks McEwen. These are all names that you and I know. Some of the younger people may or may not know them, but they were clearly
Giants,
giants.
giants in the field upon whose shoulders we clearly stand.
Right.
Brooks McCune brings back a wonderful smile to my face. I have such warm recollections of both of those giants, but Brooks is somebody whose name has unfortunately faded a little bit into history. So for all of our listeners, if you're interested in reading some amazing work, go back and look up Brooks McCune.
Yes.
Everybody knows Robert Mockemer.
Yeah, and so and at that time I had spent when I went back to Duke, I'd spend a lot of time with Gene Dewan, who I'm sure all the listeners do know. Nobody wanted to work in Gene's lab; everybody wanted to work with Dr. Markham, so it was sort of easy to get into his lab. Now he wasn't working anything on bioelectronics, but he was flexible enough to allow me to work in his lab. And then, Glenn Jaffe was there, and so a lot of other people that, so Duke was really a powerhouse. But at that point, Gene Dewan had decided to move to Wilmer Eye Institute. And so Wilmer was also strong. Julia Haller was there, Peter Campicero. From a project standpoint, it made more sense to move to Wilmer for my vitreoretinal fellowship. Actually, it was medical retina 1 year with Neil and Susan Bressler. Dan Finkelstein was there back then, and then to the second year was vitreoretinal surgery with Jean, Julia, and Peter. So I decided to do that, and it allowed the project to continue, and we were actually able to do some more early preliminary human testing with electrical stimulation before any device was ever built. And those years were very, very important. And so I decided to go to Wilmer, and I stayed on faculty at Wilmer before moving out to University of Southern California.
Right. And if I recall, the Mad Lab, Gene's lab, I think that's when we first met, right when he opened it at Hopkins, was just the time that you and I were both looking at where to land. And I love the culture that Gene built in that lab, the culture of innovation and open-mindedness and disruption. Talk a little bit about the cultural innovation principles that you took away from working with Gene in those early years?
Well, Gene was, one of the, very few people who would, was basically open to most anything, any idea anyone had, and if they wanted to pursue it, there was room to do it. Beyond that, of course, everything else you had to do. If you picked a project like I did, there was very little knowledge or support. So you had to basically sink and swim on your own. But at least he gave you the opportunity to do so. It was very encouraging, and he was just a person you could talk to. In those days, it was just easier. He was junior faculty, and you could spend a lot of time with him. And so I think that was key, is just really, just allowing people to do any and everything in the lab, and see where you go. And that was really refreshing. And a lot of other labs, you basically you kind of was you were a cog in a wheel. So there were a lot of activities going on. There were a lot of programs going on, big programs, and you would have to fit into those programs. With Gene, there were some programs going on, but you could always introduce something completely brand new, and it would be perfectly accepted.
I can't remember what the acronym stood for, the Mad Lab. What did it stand for?
Oh,
Can you
it's kind
remember?
of, yeah, yes, I do. But it's kind of interesting because it's sort of it was picked for a number of reasons. It was Microsurgical Advanced Design Lab.
That's it.
But it was back then. There was this comic,
That
uh,
magazine.
that magazine. So it was kind of like a little bit of a kickoff on throwback on that too. That there were these just mad scientists working in the lab, and there was that magazine. So it had a double entendre there. It stood for what I just mentioned, but it also had that sort of crazy mad scientist feel to it, or at least the name should have invoked that in people's minds.
Well, uh, I never thought of Gene as a crazy mad scientist, but I think maybe he wanted to think that way about himself. But he's such a huge contributor, and you, of course, are a huge contributor. Let's just tick down the list of some of the items that you have brought to the clinic. One of the collaborations with Gene, I know I came on it a little bit later with the two of you, and it was a great privilege to work with you, were the small incision, microincisional vitrectomy tools. Tell us a little bit about how you conceived of and designed the what are now ubiquitous 23 and 25 gauge systems, 27 now even.
Yeah, I mean, I think it was really a team effort, and I think Gene had a huge part to contribute to it, obviously. But he would always have an idea, and then you would have to kind of refine it or make it a reality, many times, which was challenging and interesting. So we had, back then, being trained at Duke, we always worked through trocar cannulas. I mean, that's the Duke Vitreoretinal Fellowship. Dr. Mackerman was a strong proponent of that, to put a trocar in a cannula, so you would be past the vitreous base with the instruments, and you wouldn't create peripheral dialysis and peripheral retinal tears, etc. And of course, there were 20 gauge cannulas, in some cases even 19, and so and you took the conj down and you did it, and then you sewed up the sclera because those incisions were large, and then you closed the conj over it. But the idea was, okay, can you just, basically go across the conjunctiva without having to take conjunctiva down, without and then make the incision small enough so you won't have to suture it. And so we did a lot of, mathematical analysis on the flow of fluid, and the size that needed to be in it. 25 gauge was about the right size that would not allow, a lot of not allow needing to suture those sclerotomies, and not have hypotony, without those sutures in the postoperative period, and also have enough flow in the eye from infusion, and also have a cutter that would be sizable enough to go through that, to be able to cut at a certain efficiency, and rate, so it wouldn't slow down the procedure, and then the light pipe and so forth. So, I mean, there was infusion cutter light, and then there was this concept of how small can you go without needing it to be sutureless, and then the whole concept of going across the conjunctiva. So all that, sort of came into 25 gauge was the sweet spot. 23 gauge, you can do that, but you would end up with, a fair number of times where you would, end up in hypotony if you didn't end up suturing. To be fair, at that point, we didn't really look at the tunneling aspects of it, how, you go tangentially and then vertically, so you create the step incision. So that clearly did help 23 gauge. But so those are the kind of concepts we had to struggle with. And then, of course, the rigidity of the cutter, the light pipe, so forth, especially if you're working anteriorly, behind the lens, for example, or just in the anterior vitreous base, you really are torquing those instruments, and we wanted to make sure that it didn't bend or break. So a lot of thought went into that, but it worked out nicely, and Bausch and Lomb back then supported it enough to make it commercial. Because as you know, a lot of us have great ideas and we make great prototypes in the lab, but then, who's going to make it for commercial sale and really distribute it and sell it is a whole different aspect of it. And turned out that all those dots connected, luckily for all of us, because now we have, as you said, this great armamentarium of minimally invasive, armamentarium of these minimally invasive instruments to do vitreoretinal surgery.
Yeah, I don't think that some of our junior listeners here, people in training, necessarily conceptualize just how many times those systems were reiterated. And Mark you touched on the flow, the aspiration, how they have to be balanced, the struggles we had with the light pipes, the struggles we had with the handheld instruments. In those days, instruments were largely not disposable. So the idea of switching over to a single use item and having them be machined or manufactured to the specifications of what were essentially perfect instruments, but they had to be cleaned by skilled people and prepared in the OR, and the ability to manage and monitor intraocular pressure, intraocular maneuvers like the fragmentome or the cautery even became very challenging and required lots and lots of work. And so, but there's a perfect example of, Mark, a lot of your work that has completely revolutionized. Give our listeners an idea. How many patents do you have now? I'm sure it's a huge number, but how many patents do you have now? And then how many of your patents are actually on the market, not just in ophthalmology, but overall?
Well, I don't keep an active count of it, but I think maybe like 145 issued patents is what I have, which is,
145 more than most.
well,
So,
yeah. So and they're hard to, as very well. Patents are hard to get issued. There's always prior art, especially in surgical instrumentation. It's very, very challenging. Something could be cited against you from orthopedic surgery or neurosurgery, etc. So, yeah, I mean, lucky and fortunate, and work with a lot of smart guys and gals who enable us to get these patents and really reduce it to practice. And that's the fun part about it. It's one thing to have a patent, but then can you actually reduce it to practice, keep the cost of goods low enough so it can actually be commercialized? And that's the exciting part of it. I mean, a lot of them are and have been licensed and are being used in products. I don't know exactly the number. There were probably, out of the last count, was something 60 different products, but that was many years ago, so I don't know where that count stands. But I think the most important thing about these patents are that, it's more than that for us, who are clinicians and practitioners and want to make sure that they get to patients, is to be able to actually reduce it to practice, be cognizant of the cost of goods to assemble and make these, and sterilization and other issues, beyond just the ergonomics and usability. So that's obviously just the basics. You got to have that, but then you got to have these other things too.
so if you were, when you counsel your trainees now, and I know you are training people both in the engineering programs, bioengineering programs, and ophthalmology retina. When you counsel your trainees about innovation, it's a daunting path that they have to navigate from idea to proof of concept to to IP to reduction to practice. So, what besides perseverance, which is sort of the cliche, what do you tell your trainees? How do you motivate them, and how do you get them ready for what they're about to face with bringing an idea to fruition? A lot of our listeners have great ideas,
Yeah.
but they haven't done it. So, what do you tell people?
Well, I mean, I'm asked this question so often that I actually have a talk on it, and I've given it a couple times this year. I just made the talk, because I've only literally given it 2 or 3 times this year. Um, but there are two things to this. One is the definition. One is invention, and one is innovation. So an invention is something that's truly, you know, came out just truly brand new. So the
Novel.
transistor, novel, so novel that nothing exists. And typically inventions are things that take, decades to actually reduce to practice and make products out of. But they're enormously important, because without them, we wouldn't have a lot of the other innovations. Now innovation is taking something that already exists and improving upon it. So and innovations typically make a lot of money for people and companies, because they're realizable, they're more near term. So the battery transistor inventive things took many years for people to use them, but the Tesla car uses battery driven, a car, is a small gauge surgery again. Early vitrectomy, Robert Mockemer days, many years and decades to perfect that. But then small gauge surgery is a great innovation off it that has helped us and is being commercialized more readily. So it just depends on your mindset. So the first thing is, are you the type of person who really likes inventive things, like to truly blue sky and develop something brand new? Or you're more somebody who can see things that already exist and apply them in an innovative manner. So that's the first sort of mindset. And maybe you're both. Maybe you have inclinations to do both. But then you have to kind of, in terms of innovation, you have to, and it seems like most people, who see a problem, where clinicians are more interested in an innovative solution that can help more readily. And then you just have to, think about, like we just said, the steps of it. Not all ideas are good ideas, and you have to be selective. You have to be very brutal with yourself, and say that idea that I had is not good, but this idea perhaps, and you vet it, and you vet it through those aspects of, is it, how much of an unmet medical need is this? I mean, what's the market size? Secondly, is this doable? Is it cost effective? Is it going to be something that's truly going to help physicians? You got to think about commercializing it, mass producing it. You got to think about reimbursement. These are the things we often don't think about, but you got to kind of build that in. Okay, this other widget is being sold for $10 already. So how much could you sell this new widget, which is a innovation on it? So I think you have to bring all those aspects in, and really be brutally honest with yourself. Get a lot of input from smart people around you in each one of those areas to move forward. Now, it sounds daunting. So, first start off with the unmet medical need, but be disciplined about it, to develop these technologies, because often we get into this and we just fall in love with our idea, and we're not as disciplined as we could be.
Right. And where do you tell people to find the motivation to continue? Because invariably, there are speed bumps and there are mountains that become obstacles. And whatever they are, you just listed a bunch of them. And they get doubts or they get run down by the process. What do you point to as the motivation to keep going?
Well, I think you have to realize, and that's really hard, because that idea that you fell in love with, a year, 2 or 3 or 4, is not as novel and shiny as on day one. But you have to sort of realize that these things take a long time. They're not something that can happen overnight or even within the span of months or even a year or two. They just take a lot longer. We, as clinicians, have to wear a different hat because we can go in to see a patient, we can do a laser, we can do an injection, we could operate on them. We're very used to this immediate sort of intervention and gratification and improvement on our patients' lives and their vision. Um, but this is different. You have to wear a different hat. You have to have a different concept. And as Dr. Markham told me one time, which I didn't really understand at that time, he said, you have to have a slow internal time clock. And what he was telling me is that you can't be a clinician in the lab. You have to realize that these things could take a lot longer and you have to stick to them and overcome these sorts of challenges. Now, the hard thing is, Steve, is you and I know, I mean, we're constrained by reality because time is one thing, but then money is another, and you got to make sure that you have enough funding to tie you over that time while you're trying to sort these things out. So it's tough, but you got to believe in your idea. You got to stick with it, and you got to have a slow time clock.
Yeah, I will say to our audience that I've had the privilege of sharing moments with Mark, both in the OR and with patients, and obviously academically for years. And so when I see him, just that little smile when he knows, and he'll never admit this because he's far too humble, that something that he worked on for decades is now impacting a patient positively, whether it's somebody with an Argus 2 who can see, or somebody with a regenerative patch technology who can see with a bunch of stem cells on a piece of Parylene under their macula and they're improved, or whether it's even small gauge vitrectomy surgery becoming the absolute standard of care, the minimally invasive surgery, and so many other contributions. I can see that satisfaction juxtaposed with that looking back at that slowed down time clock, if you like. You track accomplishments in eras rather than, 1 week follow up with a patient, or are they attached tomorrow? It's a very different level of satisfaction. I tell that story because for those of you who are listening and you have good ideas, we want you to innovate. We want you to find the fortitude to do all of those things, even though the tsunami of clinical care could just wash over you. It's so important to find a way to invent if you can and innovate if you can't invent. And even innovation is amazing. I mean, it can be such a contribution and so satisfying. It's such a great way that you've given back, Mark. So thank you for all of that on behalf of our field. Thank you for that. Let me ask you another question. Going back, so you're at Hopkins, and you and Gene are recruited by Steve Ryan to come out to Los Angeles, out to the forbidden badlands of the West Coast, where you turn on CNN and the world is burning, but you get out here and it's really Mayberry RFD. And what gave you what sold you on coming? I mean, Steve and Gene are probably the two greatest salesmen in ophthalmic history in terms of convincing people to do things. But it was a big move for you and your family. How did you find the bravery to do that?
You're absolutely right. It was an enormous move. I mean, all my family's in Maryland. And I was at the Wilmer Eye Institute. My parents, both my brothers, my wife's family's from Virginia, so we were very East Coast centric. Gene and certainly his family, from Alabama, but nothing out on the West Coast. I think what started it all is during that period, the Argus 2 had just started clinical trials, and I was flying back and forth. The company, Second Sight Medical, back then, um, was located out in California, so I was flying back every week, back and forth across, East Coast, West Coast, and it was just getting very tiring, and it was getting to the point where I just couldn't keep it up, and I couldn't do it. So that was one of the main motivations to move out to the West Coast. Again, project driven, just like my PhD. This time, this huge career move and family move was because of this project, and because where the company was located and how pivotal that time was to get those approvals to start first in human studies, which were so critical with the full implant. Which the first implant we did was at USC, shortly after I moved out here. So it was that was the motivation, and then of course Steve Ryan, as you said, great sales person, made it easier. He came out, he was very welcoming and said, "Look, we'll provide all the resources. This is important." But then came, this whole issue. Of course, my long partnership with Gene, and he was very well established at Wilmer. He was clearly professor and co-head of Retina, and so I went to him and said, "Look, I think from how I see it, I want to move out to Los Angeles," and so it was
Can I just interrupt and say, I was at UCLA at that time, running Retina at UCLA at that time, or about to be running Retina at UCLA at that time. I think I was the newly minted chief of Retina in those years. And I love Gene and I idolize Gene and he's my friend and he's made a ton of contributions, but it's my distinct recollection that you were the recruit. How'd you navigate that? Because Gene was a giant, still is a giant in our field. And I don't think Steve Ryan ever thought he could get Gene to come, and I don't think he was settling for you. He wanted your project here in Los Angeles.
Yeah,
So
yeah.
what happened there? How did you navigate that, Mark? Because that's
Well,
tricky. Am I
yeah,
wrong? Do I remember that correctly?
you do, and it was very tricky because obviously Gene, being it was sort of being split up. I mean, doing the things with Gene were great, but the project was equally important. So it was really tough. So I just approached Gene, and as I was saying, I mentioned the opportunity out in Los Angeles, at USC, and so Gene's, and this is the thing, as I mentioned, Gene is. He lets you do whatever. And so I just thought that, he would say, okay. But what he said is something very interesting. He said, look, instead of them getting, and he used the Lakers analogy back then, he said, look, instead of them getting a Kobe Bryant or Shaq O'Neal, I guess, referring to one player, why don't they get the whole Lakers, the entire team? And at that point, I was just stunned because it was very obvious what he was saying is that let's move Mad Lab and Gene and myself and everyone to the West Coast. And so I called Steve Ryan, and he still tells me he was driving on the 10 going to the West Side. And when I told him this, and he said, he almost crashed the vehicle, because certainly this was not what he was expecting. But he clearly realized the potential, formed the Doheny Retina Institute, and got the whole team out here. So I think it was just really Gene's flexibility and his ability to see that more could be done somewhere else, even though he was truly at the sort of at the top of the heap at Hopkins. And then Steve Ryan's ability to say, okay, I'm going to try to get the whole team, instead of one player.
Well, Steve was definitely a world-changing, charismatic figure. I always jokingly called him JFK because he was just that handsome and charismatic and such a great leader, inspired such awe. And so let's shift gears because that's a fascinating story, and I'm sure that could go on for hours. But let's shift gears because the University of Southern California is a great institution. And Doheny aside, and what's navigating all of that, but let's talk about USC and the Department of Engineering for a second and what it's been like for you to be at USC, uh, because it's a great place. Not just the ophthalmology fellowship that you guys have and the residency program, and the LA County Hospital on the medical side, but the university itself is magnificent. So speak speak to us a little bit about what was in your decision matrix about the university, the engineering departments, and so on, and what you were looking at when you were considering a life-changing move like that, and how they've exceeded your expectations, because I know that the university has exceeded your expectations.
Well, USC has been a great place for me, and so there was a corollary to that whole move because of the company. There was a large National Science Foundation grant. They're called engineering research centers, and really the universities back then could only promote one program at a time from each university to get these programs, and they're 50 to $100 million. I mean, they're huge. They last, a decade, and so at Hopkins, there was no chance that I was told that this program. We were developing implantable bioelectronics, so not only for the eye, but for paralysis and cognitive impairment, that they would put that forward. But at USC with Steve Ryan and his support, they said, you will be the number one program. And at that point, he was working very closely with the then newly appointed Dean of Engineering, Max Nikias, who later became president of USC and so forth. So Steve and Max, were able to support this program and put it forth as the premier program from USC. And we won that over Hopkins, a lot of other universities. We won that grant. And from it came a lot of the patents that I mentioned earlier, a lot of inventions, not only in ophthalmology, but outside ophthalmology. And so having Viterbi School of Engineering, which is the engineering school at USC, being so, they're top 10 back then, and they're pretty much close to that. And so having that kind of resource here in the university was extremely helpful. And then the support of the top level deans, the presidents, and the provosts to promote or put these grants forward to be able to compete and then eventually win some of these large grants was also extremely important. I just found that the Southern California was more tech centered, more device centered. A lot of people may not know that, but within USC 50-mile radius, there are like 400 small companies. They're job shops. They can make anything for you. It's kind of a hub of small companies in tech, and a big area for ophthalmic tech, intraocular lenses, a lot of the surgical devices and so forth. So it's a very different environment than Baltimore. Baltimore and Washington D.C. have come along a long ways, but Southern California is a high tech and especially focused on medical instrumentation. So USC, Southern California was really helpful. And then in Southern California, of course, Caltech, which, I collaborate with a lot. And to some extent, obviously with at UCLA, we've already talked about that. So there's this incredible amount of momentum of these schools with different type of expertise that make it an amazing environment.
Right. It is truly amazing for those of you listening. It's where the military goes shopping, and it's does support a lot of entrepreneurship and venture related things. And Caltech is obviously JPL, the Jet Propulsion Laboratory, is right up the block. Caltech, UCLA, USC, UC Irvine, San Diego. It's quite the corridor. And if you look at the uh, defense labs that are out here as well, they're pretty good. And I would say, without knowing for sure, that Mark, you've had to probably have top secret clearance because you're walking into labs and implanting things that we're not allowed to know about. Over the years, I know I had to have clearance now and then to interface with some of the groups that we worked with. But it's just a very exciting place. But getting back to the university, so what I'm synthesizing from your answer is you had high level support. Your program was anointed and chosen. It was supported. There was a consistency there. So you had not only financial resources, but space resources and leadership resources and a freedom to operate that made it a fantastic opportunity. And you've taken advantage of that and really achieved over the years. Does that sort of sum up how you chose?
Yeah, I mean, Steve Ryan took a big bet and placed a big bet on it, and then also Max Nikias, the dean. And so I think that they bet on this program and bioelectronics and ophthalmology in general. They wanted to continue to support that, and it just became, and they still say it is the jewel in the crown of the medical school, and for that matter, most of USC is really truly the jewel in the crown of the university.
Well, USC has a lot of jewels in that crown. There are some really great other departments that aren't just in engineering, but certainly in medical innovation. And congratulations to you on that. Mark, when you're training fellows, shifting gears to clinical now, long history of great fellows and fellowships there. When you're training fellows, give us two or three clinical pearls that you hope that your fellows will keep with them forever. So Robert Mockemer said to you, you should have two time clocks, right? For your innovation and your invention, you have to slow down time, right? And not have instant gratification. What are some of the big synthetic takeaways that you try and impart to your fellows? I mean, obviously, look, table stakes are they're smart, they're good people, they're ethical, they've got good hands and good judgment, but what do you counsel them on? We're our listeners are now a fellow, and they're coming in to see you after 6 or 8 months of fellowship, and you're doing one of your 360 evals on the fellows, so they get to meet with a great Dr. Humayun. What are you imparting with them?
Well, I have another Mockemer saying, which I still impart to fellows, and it's a little bit confusing and can be disconcerting, is that, what he said is the first rule of vitreoretinal surgery is that there are no rules. And it takes you back when you hear that. But what he was saying there is, and this is the thing with Dr. Markham, you always had to hear and then think a lot about it, and then ask him, and then try to figure out what he really meant. And what he means by that is that each approach, each understand the anatomy of everything. He was a big proponent of that, and that's what I make sure that everybody understands, is that look at this and try to understand the anatomy of it. Try to understand where the traction is. Try to really understand anatomically what's going on. Try to really understand your instrumentation. Why? How does a cryoprobe work? How does a laser work? I mean, how does a vitrectomy machine work? Why do you pick certain types of instruments? The ergonomics of it. Really try to understand the nuts and bolts. And this is what Dr. Markham would do. He would spend time with residents and fellows, and he would just ask you these questions. Well, why did you do this, or why did you pick that? And so forth and so on. So I think that understanding the anatomy and understanding the surgical instrumentation, once you have that know-how, then you can approach each case with that knowledge and not necessarily be rules-based, meaning I'm just doing this because, my senior resident told me to do this, or I saw somebody else do it that way. You're truly thinking about it and developing your own approach. So, and that has stuck with me, and that's how I, approach most of my, and how I've done most of my cases, and that's what I try to impart to the fellows is, you could watch as many YouTube videos, and now we have iWiki and a lot of great videos, and they're great to watch, but
And Retina Rounds.
and Retina Rounds, of course.
Yeah,
But you gotta basically start with understanding the basics of instrumentation and the anatomy, and then see, what you're looking at on those videos. Do they make sense? How could you do it better? And really don't be bound necessarily by what others are doing just because that's how they've done it before.
right. And when you have fellows that are graduating or getting ready to make a career decision, whether to go into academia or private practice or now private equity or managed care, how do you counsel them on their decision matrix? What do you tell them about what to choose? I mean, obviously everybody tells their trainees you have to search your own feelings and figure out what's right for you, but there's a more nuanced conversation. What do you tell them?
Yeah, I mean, I think that the fundamentals of academia are what I can tell them about. I don't know too much about some of these other areas because all I hear is what I hear. I haven't walked the walk in those areas of private practice or private equity, but what I can tell them is basically what I know about academia. What to expect, what I see their potential. Are they, what can they do for academia? And what to expect from academia. And then, they get a lot of input about private practice from others and some of the other things you mentioned, being part of private equity, et cetera. So then they make their own decisions. But, I feel like I really can't tell them too much about those other areas beyond because I haven't just lived them. But I can tell them a lot about academia. And then they
What do
sort
you
of
tell them?
self
What do you tell them about academia?
well, I mean, I think the key thing about academics is you want to be in it because you want to do something, that hasn't been done before. I mean, it doesn't mean it has to be in the lab. It doesn't mean it could be a different way of teaching. It could be a different way of clinical flow, whatever. But I mean, ultimately, you want to do something that's truly, something that has not been done to help patients, and so you're driven by that. If you're not driven by that, and you just then you're going to get frustrated because it's there's a lot of bureaucracy in academia. You're always dealing with departments, schools, provost, the whole nine yards, and it's a big operation. It's nothing like it's so you have to. There has to be a really good reason, and the reason is do you believe in yourself that you could do something innovative? Do you have that fire in you that you want to do something innovative? You want to change the world to make our patients' visions and lives better?
Right. And I'm presuming that you tell them don't do it to please you. Do it to please themselves.
Absolutely. Yeah. I mean, this the worst thing. Yeah, they can't do it to please me or please anyone else. That'll be fickle. It won't last long and they'll be unhappy very soon. So, and I think that having said that, not a lot of people choose the academic path. I mean, now as you and I know, I mean, most people don't go into academia. Most people go into, private practice or private equity, et cetera. So, but those who do select it, I think at least we've given them a good guidance and a good view, and a good example, hopefully, that they are not then getting into it, blindfolded. They're well aware of what they're getting into.
Now, one of the topics that I like to touch on in these podcasts is, especially in our Giants of Retina series, where you have found career satisfaction. So invention, innovation, impact, super important, and succeeding in academia. But personally, where's your motivation coming from? And where's your satisfaction coming from? My guess is, and I'll just preempt you, that because I know you so well and I want you to speak to this, is giving back to society. Like I know you care deeply about giving back to society and making the world a better place, but talk about that concept a little bit and how it's fueled you and how it sustains you, because your path has not been an easy one. There are a lot of things you've had to overcome.
Right. I mean, I think that you hit the nail on the head. I think what drives me is to really, when you go to clinic, you have so many people that are being referred to you where, tertiary quaternary center, just like UCLA and other places, academic centers are. And so you end up seeing people who have lost their vision, who are lost vision in both eyes, who are blind, and so, or who are going blind, and there's nothing that can be done. And so there's nothing more powerful than that motivating me. I saw my grandmother go blind from diabetic retinopathy, so it kind of, it is something that's deeply rooted in me. When I see those patients, when I encounter them, they really feel me to go back and work harder and do things, as effectively and hopefully as quickly as we can, realizing, what the Mackimer's old saying is that, you can't rush it. But I think that's what drives me is just the clinic, the unmet medical need that I see, and then if you're able to help even one person, it makes a gigantic difference. If you take somebody who was no light perception from retinitis pigmentosa, there's nothing in the world that anybody could do for them. And then luckily, the Argus 2 and the implant, and they can see down to, 2800 or 2400 now. Those sorts of things really change. And you see the impact it has on that one person. So even one individual, I mean, if it truly makes a difference, it's profound, and it very much resonates with me.
Yeah, and just congratulations. And you know, one thing I will take issue with, and I sort of disagree with you, is the Argus 2, like you just described, unbelievable step forward, but it didn't make a lot of financial sense. You couldn't get the cost of goods, or you haven't yet gotten the cost of goods where it belongs, but you still, and to your credit, have continued to work on it and innovate it and push it because it's going to go. I mean, there's no question whether it's that implant or another implant, it's going to happen. And the sensor interface with the neural tissue is happening. It's going to happen for sure. And whether it happens now or it happens in 10 or 15 years at a level that makes it ubiquitous, it's coming. And so, I just don't want people to not try and do things because they can't get their arms around it being a business at the beginning. Sometimes it's pure science. Sometimes it's pure invention and innovation, and it forms a basis. And I look at the Argus 2. I mean, you got it approved for goodness sakes, which is
Yeah.
remarkable. I mean,
Well,
commercially available. I never could understand the numbers in terms of, the value for a hospital, which is why we never put it in at UCLA because they couldn't, but that's like the least of it. The
yeah.
most of it is like it's a talk to me a little bit about this sensor neural interface. It's just unbelievable.
Well, a couple things. One, that first of anything costs a lot, and so basically, if you made 10 Argus 2s, only one would be functional enough, have that 55 electrodes, which is the FDA cutoff to make it. So, the yield, as we call in engineering, was very low, and it drove up those costs. But now, we have a second device, which is made by another company called IntelliMicro. It's got 256 electrodes, so
All right, so
four
four
times.
times better, logarithmically more resolution, I bet.
Right, doing much better. Is going through clinical trials, and that device costs $25,000. So now, because the yield is much better, so I think we had to stick it out. And the company, Second Sight, was owned primarily by one individual, as you know, and that individual, Al Mann, lived to a ripe old age of 89. But when he passed away, since it was so dominated by one individual ownership, and ended up getting into trouble, and it was time through COVID and stuff. But I think, but aside from that, as you know, I mean, this area is very hot right now. Elon Musk, Neuralink, Neuralace just announced that within 6 months they're going to have a cortical visual implant. So for blind people, they initially have done it with people with spinal cord injury, but now they're very much focused on the visual cortex. So this area of electrodes with neurons, this interface, this brain machine interface, retina is part of the brain, is really very exciting. I think as we get high tech companies and more unconventional than the typical surgical companies that we're used to, like Alcon and Bausch and Lomb, coming into this space, it's going to make it very exciting. So stay tuned. I mean,
Yeah, buckle your
electrodes.
seatbelts. Here we go.
Yeah, I mean, here we go. I mean, I think that we'll see what Neural Lace does with their visual cortical implant. A lot of the technology they develop can be used elsewhere, for example, in the retina, even in the cochlea, spinal cord, other places. So this is a very exciting time to be in the neural interface area with bioelectronics.
Yeah, and you are a father of that field. my friend, no matter what anybody says. So you put the flag on the moon, and I'm so proud of you. And I know everyone in our field is proud of you, and everyone in our country is proud of you. And I think even President Obama gave you a national medal of achievement. Tell us what that was like going to meet the president and receiving that level of recognition.
Well, first of all, I thought it was a hoax when they called me, because it's, obviously the people who won the national medal for uh, for technology and innovation are people like Steve Jobs and Bill Gates, etc. So, and some companies too, actually, gigantic companies. So, first of all, I thought it was a hoax. But then it was real, it was true, and it was a real pleasure to meet the president. First of all, it was an enormous endorsement of the scientific community of the work we were doing, because this has to be vetted through multiple committees and so forth, to be able to be even selected, leave alone be awarded.
So you
So
mean the phone call I made to the White House had no influence?
I'm sure it did. Yeah,
I'm
Steve,
kidding.
your call was probably picked up right
Yeah, my
away.
call was the part that made you think it was a hoax. So, what was it like? Did you take the family? I mean, when your son and daughter looked at you and your wife looked at you winning that award, your brothers and family, uh, tell us about that a little bit. Was everybody
Well,
there?
you're limited, the number of people you can invite. But yeah, my immediate family, my son, daughter, wife were there, and also my parents. Um, but it was limited to that's sort of a number smaller group. But I got to speak one on one with the President of the United States in the Blue Room, which was very exciting. He hadn't heard much, as he said, about blindness. He always hears about cancer and diabetes and heart disease, so he was very interested in learning about it. And I told him about the project, and I just thought, well, that's it. And so, but right at the beginning of the ceremony, if you go back to that medal presentation, he highlights two projects, and he actually highlights the artificial retina, or the Argus 2 program. And it's really it's amazing how quick a study he was, and how quickly he and his science writers were able to incorporate that into the opening comments. Wonderful time, great time, brilliant mind. Speaking of President Obama, and just a wonderful ceremony with the Marine Guards presenting, when they present the medal to you, they play the national anthem. So it's really quite a ceremony.
Yeah, so that's just the dream scenario. And I think it's true of all presidents. When you can manage politically to get to that pinnacle, you've got to be able to incorporate things quickly and work on the fly. But it is amazing that he was able to do that for you. What a great honor, Mark. And I think it's giving everyone in our field goosebumps to know that the retina was recognized in that way. So I want to thank you very much for taking the time to share your stories with the audience of Retina Rounds and the Retina Giants podcast. Mark Humayun from uh, the University of Southern California, Department of Ophthalmology, the Eye Institute there, and the Department of Engineering Institute there. Also very closely affiliated with collaborating with Caltech and JPL and the other universities in the Southern California area. And Mark, I'm going to take the liberty of encouraging our audience to reach out to you by email or whatever way. Mark's a super friendly guy, and if you see him in a meeting, come up and talk to him. He's always encouraging of young investigators and innovators. So, Mark, on behalf of our field, thank you. And on my behalf and Pradeep Prasad's behalf, thank you for participating in Retina Rounds. We look forward to continuing the close relationship with USC. Aaron Nagel from Children's, one of your faculty members, provided a series of surgical cases early on in Retina Rounds, and they were immensely popular. And between the two of you on the biology side and the engineering side, the innovation at USC is just fantastic. So congratulations and thank you very, very much.
Thank you, Steve. Thanks for having me.
If you enjoyed this video, please visit us at RetinaRounds.com. There you can sign up for our email list. You'll get a notification every time a new video is posted. And if you have an interesting video or a tip or trick that you'd like to share, please follow the links on our website and you can upload your video there. Thanks so much for watching.